So today I'm going to do a bacterial transformation. I'm trying out a new camera, so hopefully it looks all right. The goal of today is we're going to transform DNA from a previous ligation I had done into bacteria. The point of this is that we need to use bacteria as a tool in order to store and reproduce the DNA constructs that we prepare. So I've been working on developing a particular plasmid that expresses a protein I need for my research. So I did the ligation work yesterday, and I'm hoping that I was successful and that I can test that through this bacterial transformation. So there's about 3 steps for what we're doing today. First step is the ligation. Ligation is when you are introducing a gene of interest into a vector backbone DNA. The backbone consists of an antibiotic resistance gene. In my case, it's ampicillin. So if the gene of interest is ligated into the backbone, the plasmid circularizes, becomes circular, and it's able to express the gene for antibiotic resistance. So hopefully, only clones that have the intact plasmid will survive on a selection plate containing ampicillin. So I will use my ligated plasmid DNA, introduce it to chemically competent bacterial cells, and then spread those cells over a selection plate. Now, the cells I'm going to be working with are called competent cells. Competent cells mean that they have their bacterial cell membrane compromised, essentially. And the particular ones I'm working with have been chemically compromised. So they have a high concentration of calcium ions in the solution. And when you do what's called a heat shock, you rapidly expose these cells to a higher temperature. This causes a pressure difference between the interior of the cell and the exterior, and it causes basically little pores to form. And through those pores, my DNA, which is on the exterior side of the cell, will hopefully be shuttled into the cell, where it can then replicate or be replicated. So what I've done first is, from our minus 80 degree freezer, I've gotten 2 tubes of chemically competent bacterial cells. This cell line is called NEB5-alpha. NEB stands for New England Biolabs, that's the manufacturer. 5-alpha is the cell line. These cells are specialized for high-efficiency plasmid preparation and purification. They're also good for storing plasmids long-term, but they're not designed for protein expression. We'll use a different cell line for that. So for now, I need to keep my cells on ice as they thaw. Since they're competent, they're very sensitive. You can even call them sick, meaning that if they thaw out to room temperature or you vortex them, anything like that, you're very likely to kill the cells. So you need to be gentle with them. I thaw my cells about 10-15 minutes on ice, and then I'm going to add my ligation DNA. So this ligation mixture, there's 2 tubes. One is a control for my ligation. Essentially, it's just the vector without the ligase enzyme. and then my ligated DNA, which should be the one that has my completed ligated plasmid in it. I expect the ligated DNA, or the colonies exposed to ligated DNA, will survive much better on a selection plate. So I'm going to add 2 microliters of each DNA solution to my confidence cells. The cell solutions have about 50 microliters of confidence cells in them. I'm actually going to label the tubes so I know which one's the control and which one's the ligation. So you don't want to get those mixed up. Alright. So as I was saying, I'm going to add 2 microliters of my DNA to the cells. You want to add between 1 to 100 nanograms of DNA. 2 microliters of my solutions will be about 20 nanograms of DNA, roughly. And that's total DNA. That's not specifically ligated DNA. You can transfer the cells to a 1.5 ml Eppendorf tube if you prefer. I usually do that. But you can use the cells that, or the tubes that the cells come in, no problem. Now that I've added my DNA, again, you can't vortex these to mix them. I'm just going to gently flick the tubes 5-6 times with my finger, try and get the cells to come back down to the bottom, or the solution, I should say. You can just kind of gently tap the table with them. Usually they come down. So I'm going to let those sit on ice for 30 minutes. During this time, the DNA will coordinate with the cell membrane, and then after about 30 minutes, we will do the heat shock, which expands the pores of the cell membrane, allowing the DNA to enter the cells. So we'll come back in about half an hour. One thing I would recommend is at this point, turn the water bath on, because that takes about 30 minutes to warm up to temperature. So it's best to do the DNA incubation and the water bath warming at the same time to save some time. So now my cells and DNA have incubated for about 30 minutes, and I'm at the water bath over by our sharps container. So make sure the water bath is set. I don't know if you can see, but there's a little black lines written on the side for where you should set the dials, because this step of the transformation is very sensitive to temperature. It needs to be, for this particular cell line, exactly 42 degrees. Even deviation by 3-4 degrees, you can have tenfold loss of transformation efficiency. So another good thing is just to confirm there's water in the bath, because it does evaporate over time. And you want to make sure your thermometer reads at 42. So I'm satisfied that this is at 42. Now the next step is that you need to incubate the tubes in the water bath for exactly 30 seconds. Now this is good example of why I recommend having a watch if you're going to work in lab for these kind of experiments. So, where's my hand? There it is. I like to let it get to like 45 or a minute or something. Alright, so now I'm just going to let it sit in the water bath. You don't have to fully submerge it, just enough so that the cells are submerged for 30 seconds. There's these little floaty things too if you ever need to do longer incubations, but you can just hold it for 30. That's complete. You bring it back to the ice and let it sit for 5 minutes on ice. Also remember to turn the incubator off. And that is the heat shock step. So I'm gonna come back in about 5 minutes and show you how to do the outgrowth step. Alright, so it's been about 5 minutes. The next step is what's called the outgrowth step. So we need to transfer our competent cells into a broth solution to help recover them a little bit. And so that the plasmid that hopefully is now within the bacterial cell has time to translate the proteins that endow it with antibiotic resistance. Basically, we need to build up its antibiotic resistance before we transfer it onto the selection plate. So this stage needs to be done under sterile conditions, meaning we need to have a Bunsen burner going, and you need to be aware of what's going on. So first thing is get the Bunsen burner going. The gas comes out fast in this lab, so less is more until you get comfortable with how the regulator works on these gas lines. So now that the flame is going, the idea is that the heat from the flame will cause an upwards draft of the air. This way, sensitive materials can sit below the flame, and any bacteria that's within the air hopefully will not go down onto that equipment and instead will be pushed up by the current. So we're going to be working with a particular broth called SOC broth. A little closer. SOC is a very media-rich broth, which unfortunately means it's very susceptible to contamination. Another reason to be careful when you're working under the sterile conditions. So for NAB5 alpha cells, what we need to do is transfer 950 microliters of the SOC media to 15 ml Falcon tubes. So I'm going to open the SOC near the flame and not directly put the lid under the flame because it's plastic, but within an inch or two. And then I eject the air that's inside the pipette tip away from the lid because you don't want to push whatever that air is inside the SOC broth. And then just bring up your media. These Falcon tubes should be sterile, so you don't have to worry too much about contamination from them. Okay. And again, I'm just going to gently flame the lid a little bit. Don't put the plastic directly in the flame because you don't want to melt it. but enough to kind of bring some of that air out. Next, we need to transfer the cell solutions to the optimal broth. So I'm going to transfer about 50 microliters from my transformed cell tubes now into my optimal broth tubes. This should be done near the sterile tubes. Near. You don't have to flame these. Make sure you labeled your Falcon tubes. I did that ahead of time. So I'll take these over to our cell shaking incubators, set them to 37 degrees Celsius, and shaking at 250 RPM. I'm also going to get out 2 ampicillin resistant plates and put those in the incubator to warm up to 37. We'll let it shake for about an hour. During that time, the cells will accumulate the resistance protein necessary for them to survive on the plates. So we'll come back in about an hour and do the final step. Make sure to turn the gas off. So it's been about an hour. Hopefully the cells that have the ligated DNA now have enough protein to confer antibiotic resistance. So I've taken the 2 solutions of 2 cells out and the 2 agar plates out. They're warmed up. Before I started the camera, I labeled them. So you always want to label with as much information as you can. So I have my gene, the vector that it's in, and since it's a ligation control, I just put the vector only. And the way I set up my ligation plates is I'll do 1/2 of the plate that's diluted and another half that is concentrated cells. You can set it up whatever way you want. The recommended protocol from NEB has you make a series of dilutions. You can do that if you want. I just like to do 1 dilute, 1 concentrated. So we're going to be using the Bunsen inverter again for sterile conditions. I filled this beaker up with like 100 milliliters of ethanol. And you can see there's a glass wand in here. We'll use this to spread our cell solutions. But ethanol is very flammable, so you need to be careful working with the flame and the solution of ethanol. So I'm going to start by first transferring 50 microliters of each of my cell solutions to the 1x dilute side of my plates. Now I have 1 tip. Andrew, can you toss me that box of tips right there? Thanks. Alright. So, working near the flame, open up the plate, keep the lid face up, and I'm going to withdraw 50 microliters of my control solution and put it on the 1x side. You just want to set it on top of the agar. You don't inject it into the agar. Now, to spread it, we're going to use the wand. So, bring it out of the ethanol, kind of shake off the excess, and then you just run it once over the flame to ignite it. You don't want to hold it over the flame because you'll make the wand way too hot and you'll kill the cells. So what I like to do is kind of assign one side of the wand as the spreading and the other side is kind of a temperature check. So I know that this side of the wand is going to touch my cells. I want to keep that sterile. But I also want to make sure that the glass isn't too hot before I touch the cells. So I'll just kind of gently touch this side to make sure it isn't too hot. And then with the sterile side, just kind of gently spread the cell solution around, even it out. But you want to keep it to one hemisphere of the gel plate. When you're satisfied, you can bring it up. Just run it over for a second or two over the flame, and then resubmerge it in the ethanol, and then give it a gentle swirl. You can close the plate, the vector plate back up, and I'm going to do the same for my ligation plate. Shake off the excess ethanol, ignite the wand, pull it away, let it cool down for about 10-15 seconds. Make sure it isn't too hot, and then spread the cells around. One thing you want to be careful of is after you've ignited the ethanol, never bring the wand back near the beaker of your stock ethanol. I've seen students do that. It's terrifying. Alright. So now I need to prepare the 10x concentrated cells. So what I'm going to do is get 2 centrifuge tubes out. Label them control and ligation. And I'm going to transfer the full volume over to these cells, however I need to put them. So there should be about 950 microliters of cell solution in the tubes. I'm going to transfer that full volume over. So I'm going to take these over to our tabletop centrifuge, spin the tubes of cells for about 1 minute at 500 G. This will pellet the cells down, but not too aggressively. And then I'll come back in a minute. So I've spun the cells down. Let's see if I can show this on the camera. But at the bottom of the tubes, I don't know how well that shows, there's a little pellet of cells. So what I'm going to do now, there should be 900 microliters, or 950-ish, of solution in here. I want to pull off enough so that there's 100 microliters remaining, and then I'll resuspend my cells in that volume. So I'm just going to set my pipette for 800, and then carefully pull up. And there's, I'm probably blocking it, but there's a 100 microliter indicator on the tube. Now to resuspend, I'm going to switch back to the P200. It's set to 50. And let's see how well you can see this. I'm just going to pull up some of the solution up and down gently and work that pellet back up into solution. You don't want to be too aggressive because the cells are still a little sick, but just enough so you don't see many chunks floating around anymore. You'll notice the solution gets very turbid. Okay, so that's about good. And since I already have the tip on, I might as well plate this while I got it. So I'm going to go to my vector-only plate. On the 10x side, I'm going to pull up 50 microliters and inject that. And then just like before, gotta go on, let it cool down. This plate is done. Now I'll do the same for ligation. When I'm resuspending or mixing, I like to set my pipette to like half the total volume rather than the full volume. Because if you're pulling the full thing up and down, you have a tendency to get bubbles, which just in general is something you want to avoid, no matter what you're doing. That's the plating procedure. I'll stick these back in a 37-degree incubator overnight and let the colonies grow up. And then tomorrow, if I see colonies on my ligation plate, I'll pick those, grow them overnight, and then we get into a different experiment, which would be a restriction digest conformation. So maybe I'll do a video about that. But for transformation of bacteria, this is pretty much it.